Good day. Welcome to the BSIG Webinar Series, Episode 5. I am your host, Seth Cordes of Uncommon Catalyst. I'm joined today by Rafat Zaini, research scientist at Worcester Polytechnic Institute. Our guest today is Ken Cooper. Ken received his bachelor's from Massachusetts Institute of Technology and his master's from Boston University. With a career in systems dynamics spanning over 4 decades, Ken Cooper has led hundreds of modeling efforts. While at Pew Roberts Associates, Ken pioneered the development of project management modeling, dispute resolution with systems dynamics. He also led corporate strategy work for senior clients in several industries, including aerospace, automobiles, banking, bioscience, construction, shipbuilding, and telecommunications. His clients have included MasterCard, Fluor Corporation, Raytheon, Boeing, Northrop Grumman, Ford Motor Company, IBM, and many others. Ken is a 2-time Edelman laureate for his modeling work. In 2010, he won the Systems Dynamics Society Application Award for his modeling efforts with the Fluor Corporation. In 2011, Ken turned his focus to human biology, following a lifelong dream to apply systems dynamics modeling to understanding and curing the most complex diseases of the human body. This work led to the first clinical trial ever approved in the US based on computer simulations. Ken has led the development of models of the human immune system, HIV, type 1 diabetes, cancer, and chronic traumatic enthocatopathy. Ken, please correct me on that one. I didn't practice the pronunciation. Today, we'll look at Ken's modeling work on the human immune system and discuss the novel success that he and his team have had with this approach. We've set up a chat window on YouTube. So if you're watching via that, which everybody is, please post your questions there. We'll have some time at the end to go through them on the air. With that, I will introduce Ken, or hand it over to Ken.
Thank you, Seth, and good day to everyone. It's a pleasure to be with you. Today, we're going to discuss what I believe is a momentous opportunity for the convergence of system dynamics modeling and human disease research. Now, the challenge is that every year, tens of billions of dollars are spent on pharmaceutical research. And it's almost universally recognized that the whole process is too costly. Somewhere north of $2 billion are invested for every drug that successfully makes it to market. It's too long. It typically takes over a decade to bring new compounds to the market. And too ineffective, with very low success rates. Only 1 in thousands of molecules make it to the market. And despite the impression that might be left by seemingly omnipresent drug commercials, there are only about 20 drugs approved each year by the FDA. That's 20 drugs for all ailments, all disorders and diseases, all cancers, all viruses, all autoimmune and neurodegenerative disorders, all cardiovascular disease, 20 drugs. Clearly, the challenge is large. But what if you could simulate those complex, often multi-year human disease conditions and do so rapidly, likely in a matter of minutes? What if you could test leverage points to identify, in advance of costly testing and trials, the most promising targets for pharmaceutical treatments? And importantly, what if you could test combinations of treatments and long-term treatments and extreme dosage treatments, and do that rapidly and without safety concerns via simulation, including finding effective combinations that might never be found by conventional tests and trials? In doing so, one could rapidly screen thousands of options to increase the value of specific research targets and significantly reduce pharmaceutical development time and cost, as well as improve the probability of success. We believe this is an opportunity for the modeling community, the system modeling community, to help find ways to slow, halt, or even prevent the most devastating and deadly human diseases, thus yielding immense societal and economic benefits. We'll review here a sampling of recent modeling I've led in different disease areas, starting with the immune system and HIV. as well as type 1 diabetes, cancer, and chronic traumatic encephalopathy, or CTE. It is not the purpose here to teach biology or medicine, but rather to discuss systemic diseases from a system modeler's perspective. The more we've seen in this area, the stronger the potential we see for major contribution of system modeling to medical research. Now, the stream of work I'm describing started with modeling the human immune system, and specifically its interaction with HIV/AIDS. Why start here? Well, it's HIV research over the last few decades that has provided so much scientific knowledge on the workings of the immune system. And it was with the immune system that we wanted to start the modeling of human disease conditions for many reasons I'll mention later. The scientific literature on HIV research of recent decades also provided significant empirical numerical data that supported development and validation of the model. Further, we had the good fortune to be able to work with some top HIV immunologists. And even in the early going, and I believe that's proven correct, we thought that simulating the dynamics of the immune response to HIV would provide a platform to analyze other immune disorders, such as type 1 diabetes, multiple sclerosis, rheumatoid arthritis, as well as immunologically responsive cancers. As you'll see, there is much potential there as well. And importantly, HIV is far from yesterday's news. 35 million now live with HIV, and it causes thousands of deaths every day. So why has it taken so long to, in the search for a cure for HIV? After all, hundreds of billions of dollars have been spent over the past 3 decades on HIV research in the quest for a cure. Well, the challenge is that HIV affects and interacts directly or indirectly with many different elements of the human immune system. And we learned early on that despite the understanding that the immune system is a most elegant, feedback-intensive control system, one with extensive interaction among thousands of components, the scientific community largely works in silos. The observation we heard from the start was that, by and large, the macrophage people don't talk with the B cell people. The NK cell researchers don't interact with the dendritic cell researchers. The T cell people don't work with the antibodies researchers, and on and on. And all this despite the fact that the HIV virus interacts with so many of those different components of the system. So this is a simplified diagram of major interactions among components of the immune system that we've modeled. I offer this here not to instruct, but merely as an indication of the complexity of the system and the interdependence among its many components and its interaction with their interaction with the virus. Now, could one really think that there is one special intervention, one magic bullet treatment that will correct the behavior of this entire system once it's been infected and so many of its components affected by the HIV virus? This cause-effect map occupied about, in its paper version, occupied about half of my office wall. And I circled in red the single target area of all the current treatments for HIV. All of the standard antiretroviral treatments are focused in that one little red circle, the virus's entry into the cells and its replication inside the cells. There is scant research into the interdependent operations of the entire system affected by HIV and what treatment combinations might be more effective. And this is where system modeling can add a valuable contribution. What a simulation should and can provide is the ability to look at a number of different possible interventions and combinations of interventions that may be useful in correcting the system behavior, improving the response of the immune system so that the system itself can operate more effectively to control or eliminate the virus. Now, parenthetically, we're now seeing the success of this approach with treating cancers, and more on this a bit later. To put this in context, this is the diagram that we prepared during a flight to Atlanta and the CDC, illustrating a first pass view of the HIV virus operating in the immune system. It wasn't entirely right in depicting all of the stocks and flows of the system and the disease, but it was the basis for what became the overall structure of the working model that simulated HIV and much of the immune system, and led us to ideas for new treatments. This graphic shows the architecture of the working model that we developed for the immune system and HIV. It simulates immune activities in different parts of the body, the local infected tissue, multiple areas of the immune system, the lymph nodes, the spleen, the gut, as well as the bloodstream. And this was important in order to employ much of the research literature data. Very often these are focused on measurements of elements in the bloodstream. In each area simulated, the model simulated the activity of different actors in the system: macrophages, dendritic cells, natural killer cells, CD4 T and CD8 T cells, B cells, antibodies, chemical mediators, and of course the virus itself. In each of those simulated parts of the body, there is a cause-effect structure that simulates the activation and behavior of the populations of each type of cell and the interactions among the different cell types, both the innate and the adaptive responses. We sought to validate and test the model as much as possible using research data that was documented in the scientific literature, data on the behavior of the virus and its impact on CD4 T cells, which is the primary target of the virus, was available from the many years of HIV studies. The model did a pretty decent job of replicating all of these baseline behavioral dynamics. Wanting to further build confidence in the model, we tested in the model the conduct of several historical clinical trials of different drugs targeted at HIV. The simulation model was able to replicate fairly accurately the results of the many tests, the simulation results shown in blue here, and the average or the range of the different test results shown on each graphic. The replication of those trial results gave us the confidence to begin exploring with the model other alternatives for HIV treatments, alternatives that included using the immune system itself to combat the virus. We conducted literally thousands of simulation experiments for alternative interventions. In the course of doing that, we actually identified many combinations of levers that could be pulled to drive the viral load in the simulation to or near zero permanently, as depicted on the plot on the bottom of this graphic here. There was actually no difficulty finding quite a number of theoretical solutions that would accomplish that in the simulations. Some of the theoretically successful combinations are depicted in these next few figures. On each graphic, one such successful combination is charted against the backdrop of the cause-effect map of the system. Each of these charts shows where in the immune system that successful combination pulled a lever. Some with CD4T activity, others with different aspects of dendritic cells or NK cells or CD8 T cells. The size of each of the green circles on each diagram reflects the strength or dosage, if you will, of that factor in the successful combination that's pictured in the chart. This being one such combination and a couple of others along the way here. You'll note that some combinations require more intervention targets or more circles, and some required larger strengths of intervention, larger circles. Each of these combinations produced a quote, cure, unquote, in silico. But of course, that did not mean that there are chemical compounds available to accomplish the pulling of the correct levers in any given so-called successful combination. There was but one promising combination for which we found already FDA-approved drugs approved for other uses in individual treatments. One combination that had already approved drugs that might pull the right levers that we had identified in the simulations in accomplishing a rapid reduction of the viral load to zero. Some of the existing drugs have been used for treating HIV as monotherapies. Others have been used with cancer, malaria, and other diseases. But our research indicated that they just might pull all the right levers, that the simulation tests identified as a combination that would be effective in driving the HIV viral load to or near zero and remain there even after cessation of treatment. In other words, a functional cure for HIV. I probably don't have to tell you that this was a pretty exciting stage of the work. That combination became the basis for a clinical trial conducted with a globally respected team of investigators. Our multi-month stage 1-2 trial is believed to be the first HIV clinical trial and perhaps the first clinical trial of any kind to be based upon simulation modeling results. It was also the first HIV clinical trial, to our knowledge, that took patients off of the standard antiretroviral treatments and placed them solely on the proposed combination treatment or a placebo, and then halt the treatment. Such was the hope of the scientific review board for the innovative treatment combination being tested as a possible cure. It was not to be. After stopping treatment, all of the patients' viral load rebounded over different time frames, and they were put back on the standard antiretroviral treatment, and the virus was controlled once again. Why didn't it work? Well, blood tests subsequently showed that one of the key ingredients of the tested treatment, one of those green circles, failed to do what we wanted it to do, despite the expectations of leading scientists. This was the drug that was intended to induce the HIV virus out of latency And this was a key component of the solution. Without this particular lever pulled, the trial failed to yield a permanent cure for HIV, as indeed the model predicted would be the case in the absence of successfully pulling this lever. While all patients recovered to low viral loads after resuming standard treatment, the specific drug combination that we tested was not a functional cure for HIV. Hope remains, however, as the exact combination approach we identified in the simulations is now being pursued in other HIV trials. So, did we find a cure for HIV in that trial? Nope. We did find a promising combination, and we believe we did show that system simulation can be an effective aid in rapidly getting to identify such promising combinations that can then be tested in conventional trials. Now we're going to change course here to cover a few more applications of system dynamics in modeling and understanding different diseases. And with that, we aim to convey the breadth of possibilities. And I believe this only touches the surface of what is possible. Building on the immune system modeling, we built a pilot simulation of one example of autoimmune disorders, type 1 diabetes. In that work, we drew upon a wide range of scientific literature on type 1 diabetes, or T1D. I like this particular graphic from Van Bell that summarizes the progression to T1D over the course of many years. In this case, it's the relapsing remitting diabetes. The graphic here portrays the development of autoimmunity in the pancreas, the subsequent destruction of beta cells, the loss of insulin production capability, and over a slightly cyclical pattern, hence the relapsing remitting label, the eventual offset of hyperglycemia, as well as the so-called honeymoon phase, which you see at the extreme end of the process, immediately preceding permanent hyperglycemia. Talk a little bit more about that in a moment. A simple summary of the model that we built is pictured here. It simulates the dynamics of that progression to type 1 diabetes with the development of autoreactive effector T cells, the destruction of pancreatic beta cell mass, and the loss of insulin production, and the eventual development of hyperglycemia. We use that pilot model to simulate the multi-year progression from the first inflammatory response, and then over the next many years, the slow degradation of beta cell mass, as shown in the simulation plot here in blue. In this case, the model is simulating that relapsing remitting form of T1D in which the beta cell mass drops, has a slight recovery and plateaus, and then drops again, and another slight recovery, and eventually gets down to a level in which beta cell mass and insulin production are no longer sufficient to control blood glucose, hence hyperglycemia. Take note at the late stages of the cycle that there is a slight recovery after reaching hyperglycemia. This is that really well-documented so-called honeymoon phase observed in T1D patients in which hyperglycemia is reached and then briefly seems to sort of go away. And this stems from a temporary slight recovery in beta cell mass and insulin production before dropping off into permanent hyperglycemia. This next chart, generated by the model, simulation results, plots the simulated plasma glucose levels, once again in blue in the upper right section of the graphic. It begins with the initial inflammation And then about a decade later in this base case, there's a spike in plasma glucose levels at the beginning of hyperglycemia. And then the reduction of plasma glucose levels that briefly follows that initial spike is the honeymoon phase, in which the beta cell mass has a temporary recovery. Following this is a final round of beta cell loss, and the glucose levels climb to higher sustained levels and permanent hyperglycemia. Now, what's driving that slightly oscillating downward behavior is here a battle in the immune system between autoreactive effector T cells, plotted in red here, and regulatory T cells in blue. During the developing autoimmunity, the effector T cells attack beta cells, are trying to perform their intended duty, which is controlling against an overreaction by those effector T cells. So that their action slows the destruction of beta cell mass by the effector T cells. And once that occurs, once that slowing occurs, the regulatory T cells ease up in their response, and in response to that slowing. And this in turn allows a subsequent surge in effector T cells. And so the cycle continues over the course of many years until you get to the point where the cumulative beta cell mass destruction is sufficient to reach hyperglycemia. That cycle helps explain, and indeed in this model it drives, the oscillatory behavior of the relapsing remitting form of type 1 diabetes. Now, obviously, we want to conduct a variety of simulation experiments with such a model. In addition to the base case shown in the blue plot here, spanning 20 years, we tested different factors in the character of the immune response that would lead to that earlier onset of T1D, or a much later onset, plotted in red here. Still other factors we tested yielded a completely benign reaction, such as that charted in green. One in that case, one in which hyperglycemia is prevented from developing ever. Some of those factors in the different test cases may be characteristics of individuals and their immune response that make them more or less susceptible to the development of T1D. Of course, those test cases could also lead to identifying promising targets for treatments that intervene or alter the immune response, and this would be much like what's been observed in the exciting area of immuno-oncology that we'll discuss briefly. Now, the current conventional practice in type 1 diabetes testing and treatment faces some pretty big challenges for finding better means of slowing or preventing T1D, in as much as type 1 diabetes often goes undetected until late stages. And even if it's detected sooner, the testing of treatments, conventional treatments, needs to span very long time periods. So we believe this is another good example in using simulation to investigate disease in the systems of the human body. Another theme that we've seen in all this, perhaps as a result of looking at the dynamics of disease progression in this way, is that there may be fundamental dynamics that are quite similar across multiple autoimmune disorders. Might the relapsing remitting form of type 1 diabetes share some of these same dynamics with, for example, the relapsing remitting form of multiple sclerosis? We believe there may be some important similarities among autoimmune disorders in the initial overreaction to an inflammation and the subsequent shifting balance in the multi-year immune response that drives the progression thereafter, whether it's type 1 diabetes or multiple sclerosis and some of the many other autoimmune disorders, excuse me. So are there dynamic insights into how the disorders progress and potential treatments that may be applicable to be shared across multiple autoimmune disorders, just as is being observed in new immune-based treatments for multiple forms of cancer? That is where we turn our attention next. Again, building upon the immune system modeling, we developed a pilot simulation model of tumor development and metastasis. The model simulates the growth of a primary tumor, the genetic instability that enables that, the angiogenesis that promotes the growth of the tumor, and a variety of innate and adaptive immune responses to the cancer. Now, an odd thing about the immune response to a tumor is that not only can the control mechanism of the response fail to stop tumor growth, but some of the responding immune cells actually can join in the tumor, growing its size by virtue of containing the immune cells as well. Further, these added cells can also destabilize the tumor, reducing its structural integrity. What does that mean? It means that the immune response can actually promote metastasis, allowing parts of the now less stable tumor to break off and move elsewhere in the body. Now, that very same set of mechanisms means that if the immune system is in effect misbehaving in some of the ways that it deals with tumors and metastasis, then the right interventions in the immune system, as in immunotherapy, can change that balance of behavior with a more aggressive or really less inhibited initial response and less tumor destabilizing response, thus reducing metastasis. It's that less inhibited initial response that is the subject of some new immunotherapies that are now yielding promising results in current and recent clinical trials. Immuno-oncology is, and has been for several years, the single most exciting and promising area of cancer research. Once again, with the pilot simulation model, just to demonstrate the potential insights that such a model could provide, we varied a number of factors and conditions simulated in the model to conduct a range of sensitivity analyses or what-if tests. The resulting simulations exhibited a pretty wide range of behavior in the pattern of tumor growth. In one set of cases, those labeled with an A here, the tumor is controlled with 0 or near 0 growth over the course of the 20-year time frame simulated. In the extreme opposite, those cases labeled with a B here, there is extremely rapid growth of the tumor. This, for one example, is the case with pancreatic cancer, notable for its extremely fast progression. Other factors tested yielded a somewhat delayed growth in the tumor, such as those labeled with the C in the chart. In those cases labeled with the D, there is growth, but to a level that is controlled. And in still others, growth to an oscillating level, labeled here with an E. The key point here is that with a good system simulation, we can identify what conditions and factors about the tumor and the immune response yield those different modes of behavior, and that knowledge should inform and advance our progress toward better treatments and possibly cures for cancer. A couple of observations from this early work in modeling cancerous tumors and metastasis. The preliminary work to date suggests that the dynamics of cancer development may be similar across many cancers that are traditionally thought of as different. Different in the sense that cancers are traditionally thought of as being specific to a particular location in the body, whether that be the pancreas or the kidneys or the lungs or the ovaries. So much work goes into those individual locations, another case of silos of research, perhaps, and relatively little research on the systemic behavior that may be shared by so-called different cancers. Because of that system similarity indicated in the early modeling here, it implied to us similar immunotherapies might well be effective across what are traditionally viewed as different cancers. And indeed, this is what has come about recently. Immunotherapies that improve the treatment of multiple cancers have begun being observed in clinical trials, with one recent case being just in the spring of 2018. Most of the early immunotherapies are so-called checkpoint inhibitors that alleviate some restraints on the immune response, easing that built-in self-regulating process in the immune system that controls how strongly the system responds in attacking, in this case, a tumor. Checkpoint inhibitor immunotherapies relax those controls, allowing a more aggressive immune response. This is a classic example, of course, of improving a control system's behavior. Now, one of the early checkpoint inhibitors developed as an immunotherapy, ipilimumab, which is Bristol Myers Squibb's Yervoy, was first developed as a treatment for melanoma. Research and trials are proving now that these same checkpoint inhibitors are proving effective not just with melanoma, but with lung cancer as well. This seems an important piece of evidence supporting the idea that the same immunotherapies might be effective with different cancers if only we can find just the right immune system targets to hit with those therapies. Final example of disease modeling that I'll offer here is chronic traumatic encephalopathy, or CTE, of recent National Football League fame. Much of the initial research focused on whether concussions do cause CTE, and the evidence has been strong that they do. But how could traumatic brain injuries lead many years later to the cognitive and behavioral problems associated with CTE? We're not talking about severe brain damage in initial incidents, but rather a series of more modest concussions or even subconcussive hits. Our other work in previous modeling had led us to wonder if, as in the case of cancers and autoimmune disorders, some misbehavior of the immune system might contribute to the progression of CTE. And indeed, we wondered, could the misbehaving immune activity perhaps even be involved in other neurodegenerative disorders? Now, although immune system activity in the brain is different than in other parts of the body, some fundamental control mechanisms are similar. And we examined with another pilot modeling effort the dynamics of CTE and its interaction with the immune system behaviors in the brain. We developed another pilot system model, in this case of CTE, that simulates, for one thing, what is termed as the transdifferentiation of macrophages into microglia. The microglia are immune cells in the brain whose function is to clean up damaged neurons, such as those when there is a trauma to the head, a traumatic brain injury, or a TBI, large or small. These microglia get activated. The activated microglia enter a so-called repair stage of their function, and they release cytotoxins to assist in the cleanup of the damaged neurons. So once the repair activity is done, the microglia return to a resting state. So you can imagine the stock and flow structure of the movement of those microglia around the different stages as they originate from macrophages, get activated in response to a need to clean up damaged neurons, and afterward move from a repair state to a resting state. And if there are more damaged neurons, well, they induce more transdifferentiation of macrophages into microglia, more microglia activation to a repair state, more cytotoxins released for the cleanup process, and so on. So if you have a repeated, even mild head trauma incidents, it puts that cycle into frequent or even continuous operation. So let's examine some of the simulation results from this pilot model of TBI, CTE, and the immune system response. How could it be that the response to moderate traumatic brain injuries could contribute to ongoing brain damage long after those traumatic incidents have stopped, which of course is what's observed in the development of CTE. The symptoms of CTE in the form of dementia, depression, and behavior disorders are seen among some former professional football players and other sports players with repeated head trauma many years after moderate TBIs have occurred on the playing field. At this point, it's only post-mortem that the physical effects of CTE can be seen in the brain and a definitive diagnosis reached. So what processes are occurring in the brain between TBIs and years later, CTE? On the left here is a plot of many head trauma events that we injected into the simulation model, occurring weekly over the course of several months. Spanning the time frame to about 10 years in the plot on the right, we see a growing number of neurons destroyed years and years following the incidence and the cessation of head trauma. We'll examine why in a moment. Under different immune behavior conditions tested, the simulation results displayed here show that the cumulative damage can be less, while some other conditions yield a very small amount of damage much later in time. Now, CTE is not inevitable with few head trauma incidents, as in the scenario plotted here. With just a few TBI incidents, as shown plotted in 2A in the upper left corner there, the amount of resulting neuron damage in 2B can be short-term and limited, with no growing long-term damage. The neural damage can be repaired and cleaned up by microglia, as in 2C on the lower left plot, with no significant long-term damage, or CTE, as shown in 2D. But in the simulation results from another scenario here, we see that with many more hits or TBI events, as depicted in 3A, the neural damage can continue at a fairly steady rate and eventually enter a stage of escalating growth. It's at that point that a tipping point can be reached, as in 3C, and because the pace of damage in blue exceeds the ability of the microglia to repair the damaged neurons in red, the plot in 3D, now expanding the time frame to a full 10 years, shows that the amount of damage that's been growing since that tipping point was reached eventually becomes significant in terms of impact on the brain many years later. And that manifests itself in observable behavioral and cognitive symptoms caused by CTE. Now, in 2018, 3 years after these simulations were performed, Boston University, home of the preeminent CTE research center in the world, announced research findings that an accumulation of mild TBIs, not just concussions, can cause CTE. This was viewed as really as a huge advance in the understanding of the development of CTE, given that almost all prior research had focused on CTE as a consequence of more severe concussions. I'll note parenthetically here that in addition to the simulation-based observations about mild TBIs likely causing CTE, a logical inference from the simulation work was that, is that CTE could well be triggered earlier in life than simply among adult pro sports athletes, perhaps from any activity that includes many subconcussive hits to the head, but that has yet to be proven by conventional research. In the simulations as to why those mild TBIs can yield CTE many years later, the modeling shows clearly that very slight imbalances in the, the control function response of microglia would cause some collateral damage to neurons beyond those damaged by the trauma. When that happens, it triggers more activation of microglia, more collateral damage, and on and on. This proceeds in a snowballing, self-reinforcing process of damage. Damage in the attempted cleanup, and then more damage. And only years later would there be sufficient neural damage accumulated to generate visible cognitive and behavioral symptoms of CTE. A final footnote on CTE, those very same misbehaving microglia were identified 2 years after this modeling work in a major medical breakthrough announced by Harvard researchers in MIT's Technology Review. They called microglia, quote, rogue immune cells, unquote, that are likely responsible for the progression of Alzheimer's and perhaps even other neurodegenerative disorders for the same reasons that the model simulated for CTE. So, just as with cancers and autoimmune disorders, it seems possible and perhaps likely, excuse me, that there is a commonality in the immune system dynamics of CTE that's shared with other neurodegenerative disorders. Concluding here, we believe that it's useful, even important, to add system simulation modeling to human disease research for several reasons. First, the prevalence of feedback-intensive systems in the body and their interaction with systemic diseases and disorders. Second, the need for a systems view, not just silos of research. Third, the need to analyze dynamic behavior over long periods of time and to do so rapidly as simulation provides. Fourth, there's great value in testing sensitivities of different factors as to how they affect the workings of the system and the impact of different intervention options. Fifth, importantly, the need to test combinations of conditions and possible interventions rather than just a single magic bullet approach. Sixth, the importance of foreseeing unintended consequences in the behavior of these complex systems and disorders and unexpected side effects stemming from different potential treatments. Finally, the need to examine alternative hypotheses of how human systems and diseases work, because we simply don't know a lot of things about the workings of human systems. The ability to test in silico alternative theories and hypotheses about how those systems work would be valuable. From the early system modeling of chronic disorders, there's a theme of commonality of dynamics that emerges time and again, suggesting that there are important systemic similarities excuse me, in disease dynamics, and possibly in the nature of effective treatments and cures. Also, is it possible, and we think yes, and the world of immuno-oncology thinks yes, that understanding human system dynamics helps inform how to influence the body's own control mechanisms and systems to help slow, halt, or cure diseases? We believe the key message here is the huge impact potential for combining the work of those who practice system modeling and those who are engaged in systemic disease research. Now, that's a convergence that would have immense societal and economic benefits. Glad to exchange ideas on this very important topic. My contact and email information is shown here. Thank you again very much for this opportunity. Seth, over to you.
Thank you, Ken. That was fantastic. I really enjoyed that. So in answer to as we look at your closing question there, have you given thought as we wait for questions to roll in? And I'll take a moment of order here, please. To the people who are watching live, please do write your questions in the comments on YouTube, and we'll see if we can get those answered here live on the air. So Ken, how have you given some thought to how we do push this forward? What do we need? What's the call to action?
Well, one step is this platform that you have graciously provided to put that, put that idea and question out there, welcoming all ideas. One of the things that I'm with several others in the System Dynamics Society working on now is a platform through which we can encourage the development of human disease modeling within the ranks of the society members. The current thought is to engage in some form of, as a start, a cancer modeling challenge, a call to arms, if you will, with awards and prizes and hopefully some very prestigious scientific support. But we're in the very early stages of this. This is a really in its embryonic form. And I think the important thing right now is to get the discussion going and hopefully to pique the interest and enthusiasm of those who are in a position either on the side of the current medical science field or in the modeling world, or perhaps those who have a foot in both worlds, to reach across and help bring about some early examples of this very valuable modeling.
Okay, excellent. So we do have one question that's come in. So I want to thank you for the inspiring presentation. What is your recommendation regarding who should be the ideal client for these models? Pharma manager, clinical researcher? What are your thoughts on the intended audience?
Well, I think given my background, my inclination always drifts quickly to ensuring that there is a commercial paying client that's willing to pursue the work, and so I think pharmaceuticals represent a tremendous opportunity, market opportunity. That's not to say that that research funding through a number of different agencies wouldn't also be possible, or a combination of both. But I think there's no shortage of client possibilities if only we can get the message out to them. This is something that's outside their comfort zone by and large, just as biological modeling is somewhat outside the traditional comfort zone of system dynamicists.
Mm-hmm. No, that makes sense. So a follow-up to that. So with your experience working with legal and policymakers, how would you suggest we introduce this approach to those groups of decision makers?
Well, I think that again, this what we're doing here, hopefully, is a start. It's important to be able to get to some visionaries in the realm of take pharmaceuticals as an example, and you have to talk with a lot of people, expose a lot of people, is my experience, in order to find the one or two or three who gets it and can operate as an internal champion within their organization. And this is part of the outreach that I think a lot of us hopefully will be engaged in now.
Exactly. I'm sure we have in our networks members of these communities in one form or another, or a few degrees away from those individuals who can be those champions. And I think that's an important call to action for this audience that we have right now. If there aren't some of those decision makers who've tuned in today or who are watching post fact, can you share with us the approximate effort of what it took to put these pilot models together in terms of FTE hours or dollars, or some metric to set scale in this effort?
Sure. Let's see. I haven't stopped to add it up recently, but everything that I described today, and obviously all the things that ancillary efforts that went along with it, was done with about a dozen person years of effort. Okay. on that order. So all four of the modeling efforts I described today accumulated to about that much effort.
In total, not individually.
In total.
Got it. Wow, that's a pretty good return on investment, I would say, in terms of what you've been able to foresee in other forms of research that you brought out, in terms of how those insights have come about and become more accepted within the community years later, after you've foretold them with the modeling approach.
Well, it's encouraging, I think, not so much in simply proving accurate what we had foreseen, but I think it's a strong indicator of the power that good system dynamics modeling can bring to this arena of human disease research.
Next question for you. How did you overcome, did you encounter, first, I shouldn't presume the truth of this, but did you overcome, encounter prejudices and established this is the way we do things? And if so, how did you overcome them from within the physicians and others within the scientific community, based on these analytic methods that you're proposing?
I'm smiling because we encountered almost nothing but prejudice. And I can't speak definitively to how all of them were overcome, because all of them were not. We had, interestingly, we had many who, the vast majority, who said that what we did could not be done. In fact, there were a few who, surprisingly, surprising to me, told us that we should not even attempt it. In talking with one, I'm reminded that in talking with the dean of one medical school, I asked him how they approach the teachings of how the immune system works, and his response approximately was, "Well, we're fortunate if we can teach all of the different components of the immune system. There really is no mechanism or means by which we can teach how the system operates as a whole." And of course, that's probably true. I think that whole arena is yet another possibility, the arena of medical education through the use of simulators, such as the kinds I just described, would be an enormously valuable tool for experimentation in 21st century medicine medical training.
Absolutely. No, that makes sense. And carrying on the training element, the field of systems dynamics has benefited tremendously from the work on archetypes. How would do you think there would be similar benefit to expanding that collection to include cell models as well?
Cell models, certainly. There's so many different levels on which the system modeling can occur. Intracellular models, multicellular models, whole system models of the sort that I've described today would all be useful, and one can imagine and hope for, I think, ultimately a hierarchical system of models that, just as the Human Genome Project produced such extraordinary advances, that a systems model of the workings of the systems of the human body could develop over the course of some number of years to encompass many levels of operation within the body for I think what would be an enormous contribution to medical science.
No, I like where you're going there in terms of making that information, those dynamics, that understanding available in ideally in the public domain for those to, for a collaborative, very much like the Human Genome Project view of these dynamic systems.
Exactly.
All right, carrying on, the questions are flowing in. So next question. So your approach to identifying those levers, was that optimization driven, machine learning, Monte Carlo? How did you come to identify those levers in the particular combination?
Yeah, all of those. We'd turn on the model Friday afternoon and come back Monday morning to examine the results of thousands of simulations that had been run. We had to have some organized means of examining those results as well, and that was it was useful to have that. But all of those. But in addition to the mechanistic work, some good dynamic insight goes a long way as well, just from the minds of the modelers. And so some of the experiments were guided by our own thoughts about what ought to be interesting levers to try. But by numerical count, the vast majority were automated with all of those approaches.
Okay, so kind of intuitive, seeding the simulation to look in a particular direction, and then exploring the periphery around that, as opposed to examining the entirety of the parameter space.
Yes.
Okay. Let's see. Just reading the questions here, just a moment. So the question, I'll read it verbatim here. About your consulting experience, it looked like you showed, it looked like to show behavior modes of the model early in the modeling process. Did you calibrate the models or just show rough order of magnitude?
Well, the answer to that is different for the different models, which we put together for different purposes. In the case of the immune system model that we built to examine the dynamics and treatments in HIV, we went through a large amount of data development, validation processes, which I hinted at in the slides showing the replicating documented behavior and results in clinical trials. And we kept a large encyclopedia, if you will, of the with linked data sources to every parameter in the model that we could possibly do so. Mm-hmm. So because we were looking for guidance on the clinical trial, there was a great deal of numerical validation on that case, as there could be with any of the models. The other three, for type 1 diabetes, CTE, and the tumor model, were behavior mode models that corresponded in magnitude, direction, mode of behavior to documented literature, basically to explore what kinds of possibilities there would be in examining each of those diseases.
Okay. That makes sense. Makes sense. Well, I think that's about all that we have time for. The questions have stopped. And I want to take a moment again. Thank you, Ken. Really do appreciate it. If people wanted to contact you, what is the best way to reach out?
Through email is the best bet, and that's kencoopersd@gmail.com.
Very good.
It is on the slide that is, I assume it will be available for people to see.
Yes, yeah, we can see it. We can see it on the screen now. So that's very timely and and reinforcing. So that was very good. So to the audience, if you joined late or want to review, today's episode will be available or is available on YouTube presently, and here on the Business SIG's YouTube channel. I want to thank Rafat Zaini for helping us manage the chat, and for helping to arrange guests for these series of webinars. If you're interested, to that end, if you're interested in sharing your work or know of someone who may want to present more, please do let us know, and we'll reach out and discuss that opportunity with you directly. Stay tuned to the Google group. We will announce our next webinar soon. Thank you for joining us, and please, as always, share, comment, like, subscribe, share this with your audience, with members of your professional networks, and because we need to get this information in front of the right people, and to build those relationships and put this to use. So with that, we will see you next month, and thank you for joining.